Hello. To start today, we have a crossword. The clues are all definitions of keywords that you need to know for this topic. Give it a go for about 5 minutes, but not much longer than that. Here are the answers. Did you get them right? Our lesson today carries on from last lesson, which was about monoclonal antibodies. A quick reminder, monoclonal antibodies are specific to one particular antigen, and this characteristic makes them really useful in medicine. And it's these uses that we're going to learn about today. There's 4 to learn: detecting illnesses, locating blood clots, pregnancy tests, and cancer treatment. We begin with number 1, detecting illnesses. So I'm going to demonstrate this with an example, which is that in males, their prostate cells produce and secrete an antigen into the blood called prostate-specific antigen, or PSA. So their prostate cells are making an antigen, PSA, and they're releasing it into the bloodstream. And therefore, it's quite normal for males to have low levels of PSA in their blood. However, prostate cancer cells produce and secrete PSA too, but they do this at much higher levels, and so a higher level of PSA in the bloodstream is an indication of prostate cancer. So the way monoclonal antibodies are involved is that they can be produced in a manner to make them specific to prostate-specific antigen. So we learned how we make monoclonal antibodies last lesson. These monoclonal antibodies that are specific to PSA will be able to bind to any PSA present in the blood, and they could then be detected in a blood test. Use number 2 is in detecting blood clots. If a blood clot was to form in a blood vessel and block it, it could cause all sorts of complications. Often, it's not known that a patient had a blood clot until the problem is caused. So, for example, after somebody has had a heart attack, it would then be discovered that they had a blood clot. So monoclonal antibodies can help here because they can be produced to bind to a particular antigen that would be present in a blood clot. The monoclonal antibody could be attached to a fluorescent bead or dye. So if you have a look at my little diagram, you've got the antibody in orange, and then at the end it's got a little dot on it, so that could be a fluorescent bead. The antibodies would accumulate where the clot is because they'd all bind to the antigen in the clot, and they would then be detectable under UV light. The next use we need to look at is in pregnancy testing. This one's a little bit of a longer story than the last one. So here, when a mother is pregnant, she produces a hormone called HCG. Just notice there, it's a little h, a capital C, and a capital G. You must write it like this in an exam. If you want to know what it stands for, it's human chorionic gonadotrophin, but you definitely don't need to remember that. On a pregnancy test strip, there will be mobile monoclonal antibodies, which are specific to HCG. So mobile just means that they're able to move around the test strip. Monoclonal antibodies means they're all identical and they're all specific to the same antigen. And in this case, they are specific to the hormone HCG. These mobile monoclonal antibodies are on the pregnancy test strip and attached to a coloured bead. There are also immobilized monoclonal antibodies, also specific to HCG. These ones, because they're immobilized, are not able to move. They are not mobile. These ones are located at the test strip. These ones aren't attached to a coloured bead, you'll notice. Immobilized monoclonal antibodies, specific to the mobile monoclonal antibodies, are located at the control strip. So here we've got a different type of monoclonal antibody that is not specific to HCG, but instead is specific to the mobile monoclonal antibodies themselves. Now we've understood the setup of the pregnancy test stick, we need to understand what happens once a person urinates on it. So once someone urinates on the end, the urine starts to move up the test stick, and as it does so, it takes the mobile monoclonal antibodies with it. So these mobile monoclonal antibodies will start to move up the test stick towards the test strip and towards the control strip. If HCG is present in the urine, so if the mother is pregnant, it will bind to the monoclonal antibodies, the mobile ones, then also will bind to the immobilized monoclonal antibodies at the test strip. and as they bind here, this creates a coloured line. Then the mobile monoclonal antibodies not bound to HCG, so they'll be kind of extra, they'll be surplus. These ones will move past the test line because they themselves are not bound to HCG, and so they won't get stuck next to the immobilized monoclonal antibodies that are specific to HCG. They move past and they get to the control strip, and here at the control strip, the immobilized monoclonal antibodies are specific to the mobile monoclonal antibodies, so they will stop here, and this will create a second coloured line. Sometimes you'll hear about an enzyme being present at the testing control strips to switch on the coloured bead or the coloured dye. If the lady is pregnant, the test will show two coloured lines, one line at the test strip and one line at the control strip. If the lady is not pregnant, the test will show one coloured line, not a line at the test strip, only a line at the control strip. The final use of monoclonal antibodies that we need to learn about are their uses in cancer treatment. There are actually 3 ways monoclonal antibodies could be used in cancer treatment. Have a look at these 3 pictures, and for each one, try and write a sentence or 2 to describe what is going on. Pause the video while you do so. I'm going to start with the image at the top. So this one's showing that once a monoclonal antibody has bound to an antigen on a cancer cell, they will trigger for white blood cells of the immune system to attack and destroy the cancer cell. So you often hear about cancer cells evading the immune system. They kind of hide, but in this case, the antibodies will actually kind of signal, they'll call for white blood cells, so lymphocytes and phagocytes, to come to where the cancer cell is located and to attack them. In the next image on the bottom left, monoclonal antibodies can bind to an antigen on a cancer cell, and this binding will block the site where a growth-stimulating molecule would usually bind. In this case, it stops the cancer cell receiving this signal to grow and divide, and so it stops growing and dividing. In the third image on the bottom right, monoclonal antibodies can actually be attached to toxic drugs or radioactive substances. In this case, when these monoclonal antibodies bind to the cancer cell, it means that the cancer cell gets killed by this radioactive substance or toxic drug. So traditional radiotherapy and chemotherapy causes all sorts of awful side effects. I want you to have a think about why that is, and therefore why this monoclonal antibody cancer therapy might be better than traditional radio or chemotherapy. Pause the video. The reason is that radiotherapy and chemotherapy are not specific. Once they're in the body, they don't just attack cancer cells, they attack healthy cells as well, and this is what causes the side effects. So the reason that monoclonal antibody cancer therapies are better is that they are specific to the cancer cell, which means that the toxic drug or the radioactive substance they're attached to moves and binds specifically to the cancer cells, killing and harming only them. In theory, this means that there would be no side effects. That is all of the uses of monoclonal antibodies that you need to know. What is left to do is evaluate them. So I would like you to pause this video and look for 2 advantages, 2 disadvantages, and 1 ethical consideration. I would recommend for this to use BBC Bitesize. Remember to search Edexcel and GCSE. What you should have found for advantages is that they are essentially what the uses of monoclonal antibodies are. So the fact that we can test for pregnancy, we can test for other diseases, and we can treat illnesses like cancer more specifically and with less side effects are all real pros for uses of monoclonal antibodies in medicine. The other pro is that after they've been made for the first time, production is really fast. Next up are the disadvantages. So I said before that in theory this monoclonal antibody cancer treatment would mean that there were no side effects, but actually the human body is far more complicated than that. So due to this, trials are finding that actually there are quite a lot of unwanted side effects from monoclonal antibody treatment. And then the other thing is that monoclonal antibodies, although fast to produce after the first time, are very expensive to produce. Now a couple of ethical considerations. So we learnt in the last lesson that mice are used to produce monoclonal antibodies, and lots of people are very unhappy about the use of animals in the development of medicines. The particular issue with use of mice in these types of medicine is that in order to retrieve the lymphocytes that produce the antibodies, the mouse has to have its spleen removed in an operation. Another ethical consideration is that monoclonal antibody drug trials can go wrong. So, for example, in 2006, there was a particular trial that caused organ failure, fortunately non-fatal, but it is something to think about. Should we be putting healthy volunteers at risk for these types of treatments? And that's all the content. So just make sure you've got all the notes that you would like from this video. You can pause it and go back through it as many times as you need to. And then there is a worksheet about uses of monoclonal antibodies. So select some questions and give them a go.